Hi, and welcome back to Intro to Biology for Majors, or BIOL 1224. This PowerPoint is going to cover Chapter 10 from your Openstax book, Biology 2E, and Chapter 10 actually covers cell reproduction. This should come from Module 3 of our course. So this first slide is labeled Cells Divide and Cells Die. So cells divide for growth and development of organisms. That happens with all organisms. Cell division is vital to things such as wound healing. So somatic cells are actually body cells. So from here on out in the PowerPoint, if you hear me saying somatic cells, I'm talking about cells working within our body. And when somatic cells divide, they produce genetically identical daughter cells. And we're going to dig into all of that in future slides. Okay, so let's start off by talking about somatic cells or body cells, like I mentioned in the last slide. So actually, the process of mitosis produces genetically identical daughter cells from one parent cell. And we do have a future slideshow and chapters that cover both mitosis and meiosis, but for now, we're just going to talk about mitosis. So in our bodies, mitosis produces genetically identical daughter cells. So the only cells not in this category that are body cells are reproductive cells. So that would be the egg for females and the sperm for males. So cell division for reproductive cells in meiosis. So down here on the bottom, you have an example of a somatic cell. Here on the left, this is going to represent a body cell, and then you have a gamete cell here on the right. This is just going to represent either an egg or a sperm. So if you look, the difference is that the somatic cell has copies of both chromosomes. The gamete cell has only copies of one. So the egg will have copies of the mother or the female's genetic material, and the sperm will have copies of the male's genetic material. And then whenever they come together, they form the double chromosome we're used to seeing together. So let's go ahead and talk a little bit more about it in my notes. So somatic diploid cells in humans involve the body. For example, nerve, skin, liver, and brain. These cells contain 2 copies of every chromosome. So we'll get into this more, but that is considered what we call 2N. So a somatic cell would be 2 copies, which we would call 2N. And then a gamete cell, or an egg and sperm, they would be 1N. So they would only have 1 copy of the chromosome that we're used to seeing, and they're haploid cells. They only, once again, contain 1 copy of the chromosomes. We call them 1N. So somatic cells have 2N chromosomes that are in pairs. Gametes have 1N, or just half of that number, and they are not in pairs. So let's talk about the cell life cycle. So a fertilized egg, or a zygote, divides by mitosis. So the fetus grows and develops into a mature adult consisting of countless cells with identical DNA. So I kind of picked a photo that would show a very basic example of that. So on this side here, we have sperm. These are considered our germline cells, right? So this would be a 1N germline cell. It's a gamete cell from the male. It just has his DNA. And then we have the ovum, or the egg, which is a 1N gamete cell that just has the female or the mother's DNA. They come together at fertilization. They form somatic body cells within the fetus, which then becomes the adult person. And in this case, it's a male. And so this person will also have his own sperm that are his germ cells that go on to the next generation. So that's how that works, the life cycle in that gamete cells. So each mature individual produces sex cells by another form of cell division called meiosis. So mitosis, once again, is what we use to fertilize and to multiply this cell to become a fetus. But meiosis is what we use when we're going through cell division, which we'll cover later on. So sex cells, once again, which would be germline cells, the sperm and the egg, they combine at fertilization. So when the process of cytokinesis finishes, there are actually 2 new cells, each with a complete set of chromosomes identical to those of the original cell, and then that is when mitosis is complete. And so we're going to kind of hit on that and really dig into mitosis in the next couple of slides. So let's talk about the roles of mitosis. So mitosis helps with growth and development, repairing of tissues, and also in some cases regeneration of lost body parts. Obviously that does not happen with us, but for example with lizards who lose parts of their tail and then can regrow their tail, that regeneration of that lost body part takes place through mitosis. So for us in particular, mitosis involves the cells of our bodies, so our somatic cells. Some organisms actually can reproduce asexually by mitosis. This is not what we do. This is not what normal, animals do that we think of. So this actually happens asexually, means that they do not require a partner. So basically the organism will go through the process of mitosis to replicate its own DNA and basically come up with an exact copy or clone of themselves. And we'll get into that in subsequent slideshows, but let's go ahead and talk a little bit more about mitosis. So the goal of mitosis is to make sure that each daughter cell, which is the daughter that comes after the parent cell, right? We have a parent original, and then we have most of the time 2 daughter cells, and in mitosis we get those 2 daughter cells. We want to make sure that they get a perfect full set of chromosomes. So cells will replicate their DNA before the process begins of mitosis, and we talked about that in the previous slideshow. In chapters 14 and 15 we talk about DNA replication. Cells replicate their DNA before the process begins. Transcription will yield a messenger RNA, and then translation will yield a protein. So we talked about going from DNA to mRNA, which is messenger RNA, to then proteins, and through the processes of transcription and translation. Okay, so let's talk about that asexual reproduction through the process of mitosis. So some bacteria actually divide by binary fission. So in prokaryotes, which are those simple-celled organisms we've discussed, reproduction occurs by what we call binary fission, which is an asexual process that replicates DNA and distributes it to 2 daughter cells. And these, like I said in the previous slide, are clones of the original parent cell. So let's go ahead and look at this image and just kind of break that down. So here we have a parent cell. We have the blue ring here is your chromosome. We have a cell wall, that cytoplasm, that jelly that fills in the cell, and then we have a cytoplasmic membrane. So we have replication of DNA, which we talked about in the last slideshow, though. So that chromosome has doubled. So it goes through DNA to transcription, which then leads to mRNA, which then leads to translation, which then leads to protein. So we talked about that in the last slideshow. So we have the DNA replication. We now have 2 full sets of chromosomes. Then we go through what we call segregation of DNA. So they move apart, and a septum forms here, which is just a little area within that original cell that kind of starts to break apart. And then we have 2 identical daughter cells. So the cell is splitting into 2. Now these cells, the daughter cells, are exact clones of this parent. So that's what binary fission provides. So this is the process of mitosis taking place so that this bacteria can asexually divide by binary fission rather than needing a partner. So prokaryotes have 1 circular chromosome, which you can see right here in blue. As the cell prepares to divide, DNA will replicate, like I said, and attaches to the cell membrane. As the cell prepares to divide, using that septum that it creates here, DNA will replicate and attach to the cell membrane again, and then the identical daughter cells are formed here. Okay, so you get 2 clones of the original cell whenever you have binary fission. So this slide kind of digs more into chromosomes and what they are. So the DNA in the cell condenses into visible chromosomes. These chromosomes are highly condensed DNA wrapped around proteins. Cell division occurs when DNA has condensed into chromosomes. So for a prokaryotic cell, which would be those simple cells like bacteria, the bacterial chromosome, like we said in the past slide, is a singular circle here. We have other plasmid DNA, but this is really what we're trying to copy and make a new copy of for like binary fission, for example. So in a eukaryotic cell, so in one of our cells or a more complex cell, we have nuclear DNA that's found within this nucleus. It's membrane-bound nucleus that we look for in a eukaryotic cell. And our chromosomes look almost like an X, which we're used to seeing and we'll kind of see in subsequent slides from here on out. So in eukaryotes, DNA is in the nucleus and it's packaged into chromosomes, which then condense during the process of prophase, which we're going to talk about later. And in eukaryotes, chromosomes are complex, condensed structures that contain DNA. When the structure unwinds, the DNA helix is then visible. So DNA within the prokaryotic cell and the eukaryotic cell are different in that the bacterial chromosome here is just a circle, and then in the nuclear DNA, we have a much more complex system. Okay, so let's talk a little bit about how human body cells or human somatic cells will package DNA. So here we have example, we have 46 chromosomes, 23 pairs of chromosomes, and we have 22 autosomes. So one pair of sex chromosomes, and we are considered diploid, which means that we have 2 copies of every chromosome in our bodies, but our sex cells or our gamete cells are not. They are considered haploid, which means they are 1N rather than 2N, and they only have 1 copy of every chromosome. So a zygote is formed from the combination of gametes, each with N chromosomes, so each with just 1 N. So in this image I have here, we have the process of fertilization. So here's your sperm, which is 1N haploid, it has the male DNA. Here's your egg, which is 1N haploid, has the female DNA. Through the process of fertilization, where this arrow is, we get a zygote. So this is when we actually have the 2N DNA from both parents that's created this new zygote that will then become a fetus, right? So, as a result, the zygote contains twice as many chromosomes as the original 2 gametes, so it is considered a 2N diploid zygote, rather than a 1N egg or a 1N sperm. So, chromosomes divide during the process of mitosis. Each copy of a homologous pair of chromosomes originates from a different parent, so the genes are not identical. The variation of individuals within a species is due to the specific combination of the genes inherited from both parents. So that's basically a fancy way of saying that our genes are different from that of our neighbors or our friends because we have a very specific set of genes that comes from both mom and dad. So we have different genes than our best friend, we have different genes than our cousins, we have different genes even than our siblings, sometimes more even than our twins. So even a slightly altered sequence of nucleotides within a gene can result in an alternative trait. So that's why we have so much diversity in our population as human beings. That's why animals have diversity, plants have diversity. It all comes down to the way that chromosomes divide during mitosis and the way that we are made up from chromosomes coming from 2 different people that happen to be different than those of everyone else. We do not match exactly our siblings. We don't always match our twins. So it's a very specific and very individualistic process. So let's talk a little bit more about the cell cycle. So we were talking about mitosis earlier, and I was using words like prophase and metaphase. So I kind of want to introduce that here. We're going to go into the phases of mitosis even more in the next slide. But the cell cycle describes the events that occur around 1 round of cell division. So cell division being the process of mitosis. Most of a cell's life is spent in what we call interphase. And then during what we call S phase, the cell replicates its DNA. So I have this image here so that you can kind of see in red, we have an arrow that kind of points out interphase. And I would call interphase just the basic homeostasis, just a cell living its normal life, doing what it's supposed to do. And then we have the process of mitosis, where we actually begin that division. That's called the S phase. And then we have cytokinesis, where we actually have these 2 split brand new cells that are daughter cells of that original, and then we go right back. Each individual cell will go into interphase and live most of its life there. So we can kind of compare this to our lives. We live most of our lives in just a normal setting. Eventually we have our children and do all of these things within a short period of our lives, and then we go right back to our normal setting, and our children go into their normal setting. So it's kind of like that with cells as well. And once again, they spend most of their lives in what we call interphase. So let's talk more about the process of mitosis and each of its phases. So mitosis, once again, mitosis comes up with 2 identical clone cells from that original parent cell. So we have the process right here of interphase, which is where we spend most of our time. Our cells spend most of our time in interphase. So then we begin with prophase. And the process of prophase is where chromosomes will condense and the nucleus starts to dissolve. And so as we kind of move through, you can look at these images, and within the nucleus, you'll kind of see what we're talking about and what's happening. So within prophase, which would be this example, and then also late prophase right here, the chromosomes are starting to condense, and that nucleus around them is actually starting to go away. And you can see that here especially, that nucleus is kind of dissolving. So then we go into what we call metaphase. So in metaphase, our chromosomes actually align, and you can see them lining up down the middle of the cell. This actually happens. Our chromosomes line up, and then during the process of anaphase, they migrate to either end. So they actually split in half, and they migrate to either pole, so either side of the cell. Then we have telophase, where two nuclear membranes will form. And then we have the process of cytokinesis, where we actually split and have these two new cells. So in metaphase, once again, all of the chromosomes will line up in the middle of the cell and it's ready to divide. In prophase, which is the very beginning, it's the first phase of mitosis. The cell starts to break in some structures and build others up to the late stages coming up in the division. And the chromosomes start to condense and are visible even in a light microscope. So you can actually watch the process of mitosis through a light microscope and actually see a lot of this stuff happening. So in our previous slideshow, we actually started to introduce the idea of cancer and what it is. I kind of want to talk about it more and really dig into it because I know it's an interesting topic and it's something that probably hits close to home for most people. So cancer and the cell cycle. Cancer is actually the result of unchecked cell division caused by a breakdown of the mechanisms that regulate the cell cycle. So all of these enzymes we've talked about that kind of begin the cell cycle, we have all of these different processes that take place while the cell cycle is happening. Cancer is a result of cell division that continues to happen whenever there's a breakdown. So like we talked about in the last slide, they actually have mutations, which would be considered that breakdown within the cell cycle. And cancer just basically makes those mutations happen much more quickly, much more often, and this could be due to an inherited mutation or an accumulation of other mutations, maybe environmental, whatever else. So the loss of control begins with a change in the DNA sequence of a gene that codes for one of the regulatory molecules. So here I have an image of one example of cancer development. So right here we have a normal cell. This arrow would represent a genetic change, which then creates this cancer cell, which then rapidly divides into a malignant tumor. And cancer divides very rapidly, even more than our normal cells divide into a tumor. In this case, that would be cancerous. Cancer treatment and risk is this slide here. So cancer treatments include things like surgery, drugs like chemotherapy and radiation. Tumor cells mutate and divide very quickly, often developing a resistance to drugs. So normal drugs that we would use, cancers can form resistance to drugs because of how quickly they're able to generate. So when cancer metastasizes, its cells can travel in the body, can travel through the bloodstream and colonize in other areas of the body. So often, if someone is diagnosed late phase with a certain cancer, like in this image here on the right, for example, they have a primary cancer here. This cancer would have then metastasized into the liver and the lung. So cancer can basically metastasize and travel through the bloodstream into different parts of the body, where then it begins to metastasize and take place in those areas as well. So it follows the bloodstream and kind of lets off in different parts of the body and creates tumors in those areas as well. And once again, it divides very rapidly. It's very difficult to keep up with, which is, why it's so hard to not only find and diagnose, but to treat. So genes and the environment both can increase cancer risk. So a lot of us have a genetic predisposition to cancer. Things like radiation, chemicals, UV light, errors in replication within our own bodies, things that we deal with on an everyday basis. We try to avoid things that we know are carcinogens. But yeah, cancer is something that we've all dealt with. So I kind of wanted to talk about it at the end of this slideshow. And this is kind of a real life application for the process of mitosis and what it really means within our body. Okay, so here are our key takeaways from this slideshow. In humans, cellular division begins with the zygote and generates identical somatic or body cells. Most of a cell's life is spent in interphase. So then the next one is talking about the process of mitosis and its different phases. So first we have prophase, where chromosomes condense and the nucleus will dissolve. Then we have metaphase, where all those chromosomes line up in the middle of the cell. We have anaphase, where those chromosomes begin to migrate to the different poles and begin their preparedness to split. And then in telophase, we have two nuclear membranes that form. And then lastly, we talk about cancer. So cancer cells are the result of an accumulation of mutations that impact cell division. And please keep in mind, we're going to talk about mitosis and meiosis separately in slideshows in the future. This slideshow was really just to kind of introduce the idea. And I know we kind of began with the ideas of cell division and cell death. So that's why we really hit on cancer in the end. And if you all have any questions, please follow up with me. And if not, you guys can go ahead and continue on to your next slideshow.